Okay. Looks like dust in front of there. Oh, look at that dust cloud. I'm like, where is that coming from, my screen? Okay. So what did I draw here in black? What is this structure here in black? Well, that's the axon. And then the thing that widens out is the, I'm going to put AT for axon terminal. Okay? So there is a flow of electrons that basically sends what we call an action potential down the length of a nerve cell. Okay? So I'll draw that in yellow. Okay? So we have this electrical impulse that's coming down that's like, yay, I have to go tell a muscle cell to contract and do something. But can that electrical current jump to the muscle cell? No. Okay? What do we call this space right here in between the axon terminal and the skeletal muscle cell? What do we call that? So that is the synaptic cleft, or we can just say the synapse. I'm going to say synapse just because there's more room here. Okay? So that electrical current cannot jump across the synapse. So we need to rely on a chemical, which Dolan said earlier, which is the neurotransmitter. And the name of that neurotransmitter, Nathan said before, ACH, acetylcholine. Okay? Well, how does acetylcholine get released? So the yellow, which is the action potential, which is the flow of the electrons, it's electricity. Okay? It comes to the end of the axon terminal and it stimulates something. What it stimulates is a channel to open. And this channel brings in an ion. Does anybody know what ion this is? Calcium, absolutely. So this is I'm going to put VG, which means voltage gated calcium. I'm going to put Ca2+. Calcium channel. And now calcium comes in to the axon terminal. So it's a voltage-gated calcium channel. Calcium comes into the cell, and it stimulates, there you have these little secretory vesicles that are containing acetylcholine that are hanging out in the axon terminal. That influx of calcium stimulates the exocytosis, chapter 3 word, exocytosis of the neurotransmitter acetylcholine. So what's exocytosis mean?
The vesicle goes to the plasma membrane, fuses with it, and releases whatever's in it.
So it's vesicular transport to the outside. So now we have these little green dots, which are acetylcholine. Acetylcholine was exocytosed into the synapse, and it's going to move down its concentration gradient, and it's going to bind onto a channel. So I'm going to draw that channel right here in light blue. Okay? So the ACH is going to come down here and bind onto this channel. That's what that blue thing means. That blue and that green. We call this channel, does anybody know what we call this channel?
Sodium.
This is a ligand gated sodium channel. Okay? That's what this is. So this is a ligand gated sodium channel.
Does each one make their own, or are they already there?
What's that?
The ligand gated sodium.
They're located at the neuromuscular junction.
Okay, so they're already there, and they all just shuffle through.
Who's they?
Vesicles?
Yeah.
The vesicles are not going through there.
What is it, little green dots?
Those are acetylcholine. So the acetylcholine, the ACH, okay, binds onto what we call a ligand-gated channel. Does anybody know what ligand means? A synonym for ligand is substrate. What does substrate mean?
It's like I hate to use the same word to define the word, like little bridge or
Oh, cross bridge.
No.
Assumption is something that dissolves in something else? Or the
It's a chemical. It's something. Okay? It's something. Okay? So a voltage gated channel, back up there with calcium, what does voltage mean?
It contains electricity.
Okay. So a voltage gated channel is open because of electricity. A ligand gated channel is open because a molecule came and bound to that channel and changed its conformation. Does that make sense? I'm going to say that again. So back up in the axon terminal, that thing I drew in pink, that was a voltage gated calcium channel. That opened up because that yellow action potential, which is an electrical current, opened it up. Where the green is, which is the neurotransmitter, acetylcholine came and locked onto that blue ligand-gated channel, and it opened it up, essentially. And now that allows, does anybody know what molecule can come into the cell? So sodium. So sodium is going to come into the cell. So if we float back again to chapter 3, we learned about the sodium-potassium pump. It was an active transport, form of active transport. Does anybody remember what it did?
They open and close states, so when one would pass, that one would go.
Where was it moving stuff? Sodium-potassium pump.
Potassium out, sodium in.
It was moving potassium in, sodium out. But why was it active?
Because it was
It did take energy. Why, Dolan?
It was against the concentration.
It was going against the concentration. We already have a really high concentration of sodium on the outside, and we already have a really high concentration of potassium on the inside. But it kept moving them even though it was still going from low to high. So because of this, we have a really high concentration of sodium on the outside of the cell. So when acetylcholine comes over, binds onto that ligand gate and opens up the channel, where does sodium naturally want to rush? In, because it's built up so high on the outside thanks to the sodium-potassium pump. Now, if this is a normal, so I'm going to go back and label a couple things here, okay? This is the sarcolemma. Okay. What is this piece of the sarcolemma that's coming down into the cell called? T-tubule. This is a T, I'm going to put TT, T-tubule. So only at the location of the neuromuscular junction, which I'm going to circle and then I'm going to undo it. I'm going to do it in gold here. Only right here. That's the neuromuscular junction. What does that mean?
It's the area where the...
It's the area where the nerve and the muscle meet, okay? Only at the neuromuscular junction do we have these ligand-gated sodium channels because that's where the acetylcholine is. Does that make sense? Okay. So this cell, if it's just resting, it's like Sophia in the morning. What type of charge do you think a resting cell has on the inside?
Probably negative.
Negative. It's negative on the inside and positive on the outside. And we call that phenomenon being polarized. Okay? In order for a skeletal muscle cell to contract, to have an action potential flow through it, we have to depolarize this. Okay? So if acetylcholine bound onto the ligand-gated channel has sodium come in, does anybody know what the charge is on a sodium ion? Positive. So what do you think is going to happen to that negative on the inside, positive on the outside?
It's going to switch.
It's going to become positive, positive. We call that being depolarized. I don't have enough room to write it. So this, I'm going to erase the voltage-gated channel up there.
Why does it become depolarized again?
Because you have positive sodium ions rushing in.
Okay.
So this,
Becomes overall positive?
Yes.
Okay.
this becomes depolarized.
So why doesn't the outside of the cell become
Because you have tons and tons of positive ions out there.
Okay.
So the resting membrane potential, can I erase ligand-gated channel?
Yes.
So the resting membrane potential of a cell is negative 70 millivolts. When the sodium rushes in, okay, and again, is the sodium rushing in everywhere in the cell or only at the neuromuscular junction? Only at the neuromuscular junction until something happens, okay? So only at the neuromuscular junction is sodium rushing into the cell. And it's changing it from polarized to becoming depolarized. So the inside of the cell is becoming positive. Once we reach a certain number, which is positive 30 millivolts, then we call for, what does the director say when they want something to happen?
Action.
Action! Now the action potential can happen. So when the inside of the cell reaches positive 30 millivolts, then something kind of crazy happens. We have a different type of gate. It's not ligand controlled. What do you think controls it? Voltage. So I'm going to draw that in brown over here. Once we reach positive 30 millivolts, then we have tons and tons of voltage-gated sodium channels that open. And now, everywhere throughout the sarcolemma, not just at the neuromuscular junction, sodium rushes into the cell. So that's essentially how you get this electrical current to spread throughout the entire cell. Does that make sense? And what do we know about the transverse tubules? What are they doing? What are the transverse tubules doing?
Aren't they sharing it with like the other cell?
Not with the other cell. They're actually bringing, so I'll draw in yellow again. So this would be the electrical current being propagated or sent down the plasma membrane. Well, the T-tubules are literally the plasma membrane going where? Into the cell. Okay? So you now have this electrical current going around the cell and through it. Okay? If you think about this, this has to happen in literally fractions of a second. Okay? So you want the shortest path possible to be able to actually get through the cell. Okay? But there's a reason why we have these T-tubules, not only for speed, but what joins with the T-tubules? Kind of looks like this. The Sarcoplasmic Reticulum. Okay? So that's in green here. What do we call two ends of the Sarcoplasmic Reticulum plus one T-tubule? What does that make? We put three things together. Is that a trifecta? It's on top of a Z-disc. It sounds like a gang. This is a triad. So it sounds like a gang to me. So there's a triad that sits on top of, Nathan had said it, what does it sit on top of? A Z-disc. And a Z-disc is part of a what? Sarcomere. And the sarcomere is the contractile unit of the muscle cell. Okay? So why is this important, Miss Giro? What does, I'm going to erase the middle here. What does the sarcoplasmic reticulum store? Does anybody know? Calcium. So, there's calcium. I think about little old ladies need calcium, and I call this grandma's holy sweater. So, when the action potential comes into the cell, okay, what is the action potential again? It's a flow of what? No, it's a flow of ions. They are going from high to low. Okay, it's an electrical current. So when that yellow action potential gets brought into the cell via the transverse tubule, it opens yet another channel. What type of channel do you think this is?
A voltage gated.
This is a VG, a voltage-gated calcium channel. Another one.
Channels.
And calcium comes out of grandma and out into the sarcoplasm. What is sarcoplasm?
The cytosol.
No. Sarcoplasm sounds like
Cytoplasm.
cytoplasm. Okay?
The liquid.
Calcium is literally the final go signal for skeletal muscle cell contraction. And I'll draw that in just a second.
So the green tubes with the T-tubule we said were the sarcoplasmic reticulum?
Yes. And on top of every Z disc, we have a triad. And a triad consists of 2 terminal cisternae ends of the sarcoplasmic reticulum and 1 T-tubule. So I'm going to erase a little bit of the middle because I'm going to draw kind of what's underneath this. Does that make sense? Can I erase the middle for a second?
Yeah.
Oh, sorry.
Table 2, can I erase the middle? Okay.
Mr. What did you say was the
Sarcoplasm. Okay. So, again, what are those green structures again?
Oh,
Right down here. Sorry, I wasn't specific. So right here.
Sarcoplasmic reticulum.
Sarcoplasmic reticulum, and this is a
T-tubule.
T-tubule. And that structure, those 3 things make a what? A triad. So underneath, okay,
Is a T-tubule just, a transverse tubule is just an extension, right?
of the plasma membrane.
Okay.
Okay. So here's a Z disc. Underneath here would be a Z disc. Okay. What myofilament attaches to the Z disc? Actin. So I'm going to draw that in blue. So we've got actin, 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 actin. Okay. What is in the middle? Myosin. Does myosin attach to the Z disc? No. Never. Why am I making myosin look bigger? It is the thicker filament. Okay. So this Z disc that I drew on this side over here would be underneath the triad. Does that make sense? I just can't draw in 3D. Sorry. So, what ion comes out of the sarcoplasmic reticulum? Calcium. Okay? Who does the heavy lifting when it comes to I'm going to pause this for a second. So, in the green, in the sarcoplasmic reticulum, which I just made a little X over here, that's where calcium is stored. That's inside the cell, okay? The T-tubule, which is the thing that's in red that comes down, is what brings down this voltage change, this action potential. That stimulates voltage-gated calcium channels in the sarcoplasmic reticulum to release calcium. Calcium comes out into the sarcoplasm. Okay?
Oh.
What it is going to do, calcium is actually the final go signal for sarcomere contraction. So when the sarcomere contracts, what do I mean by that? The myosin comes over and binds onto actin here. The myosin comes over and binds onto actin here and pulls it towards where?
Towards the M line.
Towards the M line. Okay? Towards the M line. Well, that happens because of calcium and ATP. So, at rest, okay? At rest, actin is actually bodyguarded by 2 molecules. Those molecules are troponin and tropomyosin. So myosin cannot bind onto actin unless calcium is present. So I'm going to talk through this, and then I'm going to have some bodies up, and we're going to act it out, okay? So in a natural resting state, okay, We have two bodyguards on actin, troponin and tropomyosin. When calcium is present, troponin really, really likes calcium. So calcium comes over and binds onto troponin, and it's like, oh, hey, what's going on? And troponin moves, and it pulls its buddy, tropomyosin, along with it. When that happens, the myosin binding sites on actin are now exposed. That sounds so dirty. They're exposed, okay? Now, myosin is able to bind onto actin. However, myosin needs to get the energy to be able to bind to actin. Who do you think is going to provide the energy? ATP, okay? So we need an ATP molecule to come over and bind onto myosin, and it basically makes these little, they're called myosin heads. It makes these myosin heads like, I'm ready, okay? Once calcium comes into play, it distracts the bodyguards, so now the actin sites are free. ATP has been bound onto myosin. Myosin can grab onto actin and pull it towards where? Where does myosin pull actin towards? The M line. So if you can think of, like, I'm one big long myosin, ATP comes over and activates the little heads, okay? Calcium comes over and binds to actin, exposes the binding sites. Now myosin can grab on. Myosin can pull actin towards the M line. Before myosin can let go, another ATP molecule has to come bind to it so it can be ready again. Does that make sense? Have you ever heard of rigor mortis?
Yes.
What is rigor mortis?
Isn't it
When
one
your
of the
body's-
muscles?
So it's movement of the muscles, contracting of the muscles when you die, okay? So let's think about something. What did I just say to you? We need ATP for myosin to grab on. We also need ATP for myosin to do what? Let go. What do you think you stop producing when you die? ATP. So if there's been some muscle contraction going on, those myosin bridges are not going to be broken because there's no more ATP. So the body can continue to move. It's not going to let go. It causes these contractions to occur. Now, it's only going to be for a certain period of time because what's going to happen to all the cells in the body? They're going to start dying and decomposing and stuff like that, okay? So rigor mortis isn't going to be occurring, weeks after death. It's within a few hours after death. Why? Because ATP stops being produced. Those myosin cross bridges can't let go, okay? Now, this is a cycle, okay? So every time your skeletal muscles fire, this needs to happen, okay? So we have to go back to being polarized before another action potential from a nerve cell can come again. Well, what's going to happen? So let me erase the middle here. So what did I tell you was the number, the resting membrane value of a skeletal muscle cell? Negative 70, okay? So we went from negative 70. What ion rushed in and changed it to positive 30? So thanks to the rush in of sodium, we then went to positive 30. Once we reach positive 30, the director says action. Voltage-gated sodium channels open. Sodium rushes in throughout the entire sarcolemma. Okay? We then get to a place where we're at positive 40. At positive 40, the sodium channels close. And now we need to go back to that polarized state. What is the polarized state for a cell? What is the charge supposed to be on the inside? Negative. So thanks to the sodium-potassium pump, what ion do we have that exists in a really high concentration on the inside of the cell? Potassium. So when the inside of the cell becomes positive 40 millivolts, potassium voltage-gated channels open. And where do you think potassium is going to go? It's going to go out, absolutely. I'm going to draw that in green. So when it gets to positive 40, now potassium What charge do we have on a potassium ion? Positive. It's going to go out. And we are going to do something called repolarize. What does it mean if you say, like, oh, we're polar opposite? Very different. So now we're going to go back to negative on the inside, positive on the outside.
Where does the K plus move out of? Does it move out of the sodium, the ligand-gated sodium channels? Does it move out of that one? Is there separate potassium channels, or does it move out where the sarcoplasm?
These are voltage. They're opened up because of electricity. So you've got these voltage gated channels. In your book, they might show them as the same one. Could there be two passages going on? Absolutely. Okay. For our simplistic version of this, I want you to know it's because of the electrical change that sodium gates close, potassium gates open. Does that make sense?
So we can just draw these around.
Yes. Okay. So again, why is potassium going out of the cell?
Because it's positive.
Yes, and what is that going to do?
Make it polarized.
What number do we need to get back to?
Negative 70.
-70. That has to happen in order for another action potential for a nerve cell to fire. And if you think about this, in order for me to continue talking, in order for you to continue moving, you're having continuous signals coming. Okay? So if you think about how fast, I mean, it takes me 5 hours. It's taken me like 26 minutes to explain. However, it happens in fractions upon fractions of a second.